interfering.
Okay. Right. A very important thing in life, in biology, is the fact that the shape of a molecule is very, very important for its function. To just summarize, shape means function. So this molecule, this enzyme, is like this because it needs to be like this in order to do its job. So this is the substrate, the molecule that will react, and it goes into the active center, it gets locked in, and then something will happen. For example, it will get separated into two products. And look, it's just right for it. Now, if you change the shape of the molecule, because the amino acids change, then this substrate no longer fits. So the function is actually lost, and this protein is now useless. So, trying to teach about proteins and protein folding is very difficult because it's all very new and very just abstract for the children. And you tell them primary, secondary, tertiary, quaternary structure. Well, what's this, miss? Then you go. Primary structure is the sequence of the amino acids. So, and it really matters. One after the other and after the other in a particular sequence. So this red one here, say this is valine, this cannot be here, otherwise your protein will probably not function properly. So this is the primary structure. And then the children, the students, can get their own bits, and they can choose 20 different kinds of bits to represent the 20 different kinds of amino acids, and then they can choose loads of each kind, and they can make their own protein. And then you go on into explaining the secondary structure, and you say alpha helix. So I will go on and say, right, alpha helix is something like this, and it's held like this by hydrogen bonds. And then you say, okay, next one, beta pleated sheet. So you go like that, and you kind of shape it in a way that's visually for them okay to understand, and then hydrogen bonds keep this shape together. If you break the hydrogen bonds, then the shape is lost. And then tertiary structure. These will further fold into a shape that might look like it's whatever, but it's definitely not whatever. It's kept there by bonds. And then you get this shape that's exactly what it needs to be in order to do a function. And its function might be to keep this molecule in place in order for a reaction to happen. Now, when we finish teaching about the protein structure, we need to teach them about protein denaturation. And it's when a protein loses its function due to temperature, due to acid concentration, change of pH, and due to salt concentration and some other factors. So, to demonstrate this, it's very, very easy because you have this shape and then you can apply temperature. So, then the temperature, your protein is unfolded and it can no longer do its function, so you've lost your protein here. And you can apply it to life and say, why do you feel funny when you've got temperature, when you're real? And it's because our enzymes don't work as well as they should be, so you need to actually maintain your proteins in the right structure. And have some more time. You can go on into explaining mutations. Mutation is a change on your DNA sequence. So if a base changes in your DNA, it could alter an amino acid or more than one amino acid. And I've done these amino acids in a different color here because they're very important for the binding of the substrate. So if these amino acids change due to mutation, then it's really, really important for the protein. It won't work. This is useless. And then you go and explain that not all mutations make your protein useless, because if one in here right in the middle changes, it might not have the same effect. So it depends where in the protein it is. So they understand how mutation works. And then you can expand onto teaching carbohydrates. Carbohydrates, you can go in the classroom looking like that. And then you say, oh, Miss, what's this? Then you say, it's my glycogen. My glycogen, where is that? It's in my liver. And if I haven't eaten in the morning, which I haven't today, then my liver will be letting out glucose molecules which are connected with a glycosidic bond. And then this glucose is going to go into my bloodstream and it's going to keep me going on. And that's really important. It's a very important molecule. In plants, you have starch, and starch comes in amylose and amylopectin, so they can make these models themselves, so they really have fun making it while understanding and actually remembering about all these things you've been trying to tell them. And I have found that this way you can get the students to have fun and learn, which is really great.